As the next step in learning about human behavior and how it affects human well-being, I'd like to discuss with you some of the basic concepts regarding how the brain and nervous system work. In this presentation, I will be using visual materials from a resource at McGill University in Canada called The Brain from Top to Bottom, available at the website shown here. Before talking about how the brain works, I'd first like to discuss how the new health insurance exchange in California works. We have all read about or heard about the opening on October 1st, 2013 of the online health insurance exchanges created by the Affordable Care Act. In order to find out about various insurance plans that are available, to determine our eligibility for possible federal subsidies, and to sign up for insurance coverage under a plan, in California, we simply need to log on to the website coveredca.com. This single website can help us with all these tasks. On the surface, it appears as though we're communicating with a single computer on a single website. In actuality, we're communicating with an extensive wired network of computers, all rapidly communicating with each other so as to carry out these tasks. In actuality, when we log on to coveredca.com and enter our information, CoveredCA begins communicating with a series of other government computers. To confirm our identity, CoveredCA first communicates with the Federal Social Security Administration. To confirm that we are U.S. citizens or eligible permanent residents, it also connects to the U.S. Department of Homeland Security. Finally, to confirm our taxable income as reported on our previous year's tax return, it communicates with the IRS. As we might expect, these computer systems often communicate with each other on a regular basis. Thus, what appeared to us as a single website was, in actuality, a complicated neural structure of various websites, each networked together. The efficiency with which this network functions will depend both on how well each site is programmed and on how efficiently they can share data with each other. Our brain functions in a manner analogous to the CoveredCA website. From the outside, it may appear that our brain is a single organ that functions as a whole. In fact, our brain is an extensive network of specialized sites spread throughout the brain and wired together so as to perform different functions at different times. What makes up these various functional areas of our brain are billions of specialized nerve cells called neurons. These neurons clump together in different ways in different parts of the brain so as to perform different functions. In order for these function centers to work, the neurons also send out spindly extensions referred to as axons. The clumps of neurons are referred to collectively as our gray matter, while the collection of communicating axons are the white matter. Looking at this cross-section of a human brain, as it appears on the website Big Brain, we can see that some of the brain appears to be darker gray, the gray matter, while other parts are a much lighter color. Thus, we have the terms gray matter and white matter. To understand how the brain forms its various functional parts by collecting together billions of different neurons, we need to go back to the role of the stem cell in early development of the embryo. In its early stages of division, the embryo begins to develop different types of cells through the activation of different segments of DNA. A very early step in this process is for certain stem cells to become specialized as neural stem cells. As they rapidly divide and redivide, these cells differentiate into cells that form neurons and other cells referred to as astrocytes that provide a range of functions that support the neurons. As a neural cell goes through the process of differentiating into a functional neuron, it develops three different components: the body of the nerve cell, multiple short extensions called dendrites, and a long, thin extension called the axon. At the tip of the developing axon is what is referred to as a growth cone. The growth cone has multiple small projections that sense chemical signals in the developing brain. These chemical signals are created by other cells to which the axon needs to connect to establish the required linkage between neurons. As the axon finds and connects with the target cell or cells, it then sets up a communication system by exchanging different chemicals, referred to as neurotransmitters, with those cells. Where one nerve cell connects to another is called a synapse. Communication between nerve cells occurs when a stimulation is conducted through one cell to its synapse with another. When that stimulation reaches the synapse, it triggers tiny bubbles in the tip of the neuron that contain neurotransmitters. These bubbles are referred to as vesicles. These vesicles then fuse with the outer membrane of the neuron, and in doing so, release their neurotransmitters into the synaptic space. The neuron on the other side of the synapse senses the presence of these neurotransmitter chemicals and in response propagates a new stimulus to be carried along its length to where it makes other synaptic connections with still other neurons. At this point, I'd like to stop and acknowledge the groundbreaking work done by a team of scientists for which they were awarded the Nobel Prize in 2013. Thomas Sudoff of Stanford University was one of these prize-winning scientists. These researchers were able to identify the chemical components that are essential to the membrane fusion process that allows the vesicles to release the neurotransmitters they contain. As described in the Stanford News Service's announcement of Professor Sudoff's Nobel Prize, Sudoff purified key protein constituents sticking out of the surface of neurotransmitter-containing vesicles, protruding from nearby presynaptic terminal membranes, bridging them. Then, using biochemical, genetic, and physiological techniques, he elucidated the ways in which the interactions among these proteins contribute to carefully orchestrated membrane fusion. As a result, synaptic transmission is today one of the best understood phenomena in neuroscience. Congratulations to Professor Sudoff. The ability of one neuron to rapidly transmit an electrical impulse from its cell body along its axon to the synapse, where through the release of chemical neurotransmitters it is passed on to the connecting cell, is what allows the brain's various functional components to communicate with each other. Perhaps not surprisingly, the speed with which a nerve impulse is transmitted along an axon to the synapse is crucial in determining the effectiveness of the impulse transmission. This is where a myelin sheath comes in. In order to facilitate this transmission process, the brain has scattered throughout its tissue specialized cells called glial cells. When these glial cells sense through chemical processes that an axon is regularly transmitting electrical impulses, the glial cells wrap themselves around the axon and produce a sheath of a fatty substance called myelin that surrounds the axon. When an axon becomes myelinated in this way, it is able to transmit its impulses many times faster than without the myelin sheath. Interestingly, when an axon for some reason stops transmitting impulses on a regular basis, it often loses this protective myelin sheath in a process referred to as demyelination. Through this process of the clumping together of neurons with a similar function, connecting these neurons with other functional areas of the brain through axons closely aligned with each other, and integrating these various areas together, our brain is able to produce a unified response to an external stimulus. This illustration shows the various parts of the brain involved when we develop and perceive an emotional response to a stimulus. These parts work collaboratively to develop a coordinated emotional response in much the same way as the covered CA website provides a single coordinated response to someone seeking to sign up for health insurance under the Affordable Care Act. In this way, the brain develops various different sections, each involved in different yet coordinated parts of the mental process. As this picture illustrates, our brains have anatomically different components. The largest part of our brain is our cerebrum, which contains a number of different sections, principal of which are the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. Tucked under the occipital lobe is the cerebellum. Within these lobes are specific functional segments. For example, the area referred to as the primary motor cortex and the adjoining area 6, shown here. As shown by the shadow behind the picture, our brain also has two different halves, one on the left and its mirror image on the right, referred to as hemispheres. These hemispheres communicate closely with each other but often perform different yet complementary functions. There is another very important part of the brain not shown here, the amygdala, thalamus, and hypothalamus. These sit under the hemispheres in the central portion of the brain. I'll discuss these areas in a later lecture. This picture, taken from an online learning resource provided by the publisher Prentice Hall, illustrates which areas of the brain control many of the key functions we carry out on a day-to-day basis. Our brain has become highly specialized, with different areas performing different specialized functions. One additional area of the brain that I should mention at this time is the basal ganglia. As illustrated in this picture, the basal ganglia are tightly clumped groups of neurons that are located essentially in the center of the cerebral hemispheres. Perhaps not surprisingly, they have a central role as a communications hub, receiving and relaying messages between different parts of the brain, especially when physical action is part of the response to external stimuli. Before moving on to consider the various parts of the brain that are involved in activities such as language and reading, I would first like to identify two particular areas of the brain that play key roles in receiving sensory inputs. The first is the visual cortex. This is an area located on both sides of the brain at the very back of the occipital lobe. Nerve impulses generated by the eyes are transmitted to the visual cortex, where the shape, size, and color of what our eyes perceive is registered. Another important area is the auditory cortex, located at the junction of the parietal and temporal lobes. Nerve impulses generated by the inner ear in response to sounds picked up by the eardrum and transmitted through the middle ear and sensed in the auditory cortex. Immediately adjacent to the auditory cortex is an area referred to as Wernicke's area, which is involved in interpreting the sound patterns we perceive. Now let's move to the crucially important human behavior of language. This picture illustrates the various steps in language using the example of a mother and her son talking to each other. As the mother speaks, the sound waves are picked up by her son's ears, converted to nerve impulses, and transmitted along the axons to the auditory cortex in Wernicke's area. Once he makes sense of what he has heard, he then transmits this information across a tightly bundled group of axons referred to as the arcuate fasciculus to an area of the frontal lobe referred to as Broca's area. Here he comes to an intellectual understanding of the meaning of what has been said to him. In this case, the son, after understanding what his mother has said to him, wants to respond. He does so by sending additional impulses from Broca's area to the motor cortex, which then transmits impulses to the muscles of his mouth, lips, and throat in order to form words that the mother understands as language in response to the words she has spoken. Now, let us look at another example of a neural circuit, this time the one involved in reading. In this case, reading something out loud. Our eyes quickly view the images of the letters on the page we are reading. This sensory stimulus reaches the primary visual area where it is decoded and recognized as the visual image of the letters. The resulting information is then sent to Wernicke's area, where we link the visual image of the letters to the sound of those letters that they make when they are spoken. This combined visual and auditory interpretation of what our eyes have seen is then transmitted to Broca's area, where we compare it with similar information we have already stored in memory based on having encountered these images and associated sounds repeatedly in the past. If we are reading silently to ourselves, the information stops here with us having interpreted and understood what we have read. If we choose instead to read out loud, there is a further step of this information being conveyed to the motor cortex and from there to our oral structures where they come out as spoken words. As we might imagine, the better we are able to transmit this information across the arcuate fasciculus, the better we will be as a reader. This is precisely what Yeatman and colleagues were measuring in their study of 7-year-old children and their comparative reading skills, which we discussed in class. As shown here, over time, the better the conduction over the arcuate fasciculus, this was associated with increased myelination of the axons in the fasciculus, the better the reader the child was. Learning new words when spoken or in reading involves what is referred to as brain plasticity. I would like to read to you how the scientists at McGill University's project on the brain from top to bottom describe this process. They say, every time you learn something, neural circuits are altered in your brain, thus facilitating the passage of nerve impulses along a particular circuit. The pathways along which information travels through the neurons of the brain can be compared with the paths through a forest. As people keep taking the same route through a forest, they wear out a path in it. And the more people who take the path, the more deeply it is worn and the easier it becomes to follow. The same goes for our memories. The more we review them in our mind, the more deeply they are etched to our neural pathways. To learn a new word, you repeat it to yourself several times, and this selects and strengthens the connections among these various circuits in your cortex. And it is this new, durable association among certain neurons that will form your memory of this word. All your memories of events, words, images, emotions, etc., thus correspond to the particular activity of certain networks of neurons in the brain that you have strengthened connections with one another. These concepts leave us with a series of very important questions, the first of which is, when is our brain the most plastic? It would seem that infancy and childhood is when our brain is most rapidly developing, and thus when the brain's plasticity is at its greatest. We then ask, as we get older and further along in school, does our brain maintain its plasticity? It seems clear that learning continues throughout childhood beyond infancy and throughout the school experience. Otherwise, why go to school? While the degree of plasticity may slow down as children get older, it certainly does not stop. Given the opportunity, children who initially were slow readers and slow in language development can still learn new concepts, new ideas, and new words. Third, if by adolescence we have not developed certain key neural pathways, for example, reading, is it too late to do so? Even if adolescents have not developed the neural pathways to be good learners and good readers, they still have the potential to do so. It may take more work, but the brain maintains its plasticity throughout adolescence. Finally, can we still learn new things as adults? Of course, the answer is that learning and associated brain plasticity do slow down with aging, but they do not go away. It's never too late to learn new ideas and concepts.